Let me also turn on my camera while I'm at it. Hi, everyone. Okay, here we go. Hi.
And I will just, so this is conditional, Timothy Barry presenting conditional resampling improves calibration and sensitivity in single-cell CRISPR screen analysis, joint work with Suran Wang, Catherine Roeder, and Eugene Katsevich. Take it away.
Okay. Hi, everyone. Yes, so I'm Tim. I'm a PhD student in the Department of Statistics and Data Science at CMU in Pittsburgh, Pennsylvania. And yes, this is recent work on an analytical tool for single-cell CRISPR screens. So yes, my co-authors are Suran Wang and John Morris, and my advisors are Catherine Roeder and Gene Katsevich. So I want to motivate this by first discussing what I like to call the dark genome. So we know that genome-wide association studies over the past decade or two have revealed hundreds of thousands of variants that are associated with disease. And it's been surprising to people that most of these variants are not actually located inside of protein coding exons. Rather, they're located outside of genes in non-coding regions. About 95% of variants associated with disease lie in non-coding regions. Probably they have a role in regulating gene expression. And so a central goal, I think, in genetics, over the coming decade is to shed some light on the dark genome and to understand the role of these non-coding variants in disease and other human traits. A technology that is extremely promising for making progress on this question is single-cell CRISPR screens. For those who aren't familiar, single-cell CRISPR screens are a technology. Basically, it combines CRISPR perturbations with single-cell sequencing. The idea is to use a CRISPR perturbation to target some region in the genome, and then to use single-cell sequencing to assess the effect of that perturbation on, say, the transcriptome of the cell or the expression of the genes. And so we can use this technology, for example, to map enhancers to their target genes in a very precise way at genome-wide scale. So in this little schematic here, the enhancer or the candidate enhancers, say, are the orange boxes and the genes are the blue boxes. And using these single-cell CRISPR screens, we can sort of do this very precise mapping of enhancers to genes. And so that's sort of the focus of this talk. And so just to describe the assay in slightly more detail, basically there are a few steps. So the first step is to culture a large number of cells. Maybe we have a couple 100,000 cells, and we're going to target each cell with some set of guide RNAs. And these guide RNAs will be randomly assorted into the cells. Each cell will probably receive about 30 distinct perturbations. These guide RNAs are targeting different regions in the genome. And then what we're going to do is for each cell, we're going to basically measure its transcriptome, that is to say the expression of the genes, and we're also going to measure the guide RNAs that the cell received. And then the final step is to basically do differential expression testing. So there are different ways to do this, but a common strategy is as follows. Basically, we're going to do gene to gRNA sort of pairwise differential expression testing. Consider here the orange guide RNA. What we're going to do is we're going to split the cells into two groups. We're going to sort of look at the cells that receive the orange guide RNA, and then we're going to compare those cells to the set of cells that did not receive the orange guide RNA. And then sort of for all genes in proximity to the enhancer being targeted by the orange guide RNA, we're going to test for differential expression across these two groups of cells. And if there's some gene that's differentially expressed across the two groups of cells, that's strong evidence that the orange guide RNA is targeting some, say, candidate enhancer or some enhancer that's like regulating that gene. So that's sort of like the basic idea of these sort of enhancer targeting single-cell CRISPR screen experiments. Yes, so it's a very promising technology, but it turns out that data produced by single-cell CRISPR screen experiments pose substantial statistical challenges. So to illustrate this, we look at two recent single-cell CRISPR screen data sets. The first was published by Gasparini et al. In 2019, and the second was published by Shia et al. In 2019 as well. And what we observe is that the analysis methods that were used originally in these papers to analyze the data exhibit very severe miscalibration, which is a problem statistically. So basically, we can assess the calibration of methods by examining what are called negative control guide RNAs. So the idea is some of the guide RNAs that are sort of transduced into the cells don't actually target any region in the genome. Rather, they just kind of float around in the cytoplasm of the cell and don't really do anything. So what we can do is we can pair these non-targeting guide RNAs. There are probably several hundred per experiment. We can pair these non-targeting guide RNAs with genes, and we can check for differential expression. And in this setting, we're under the null hypothesis. So there should not be any change in gene expression associated with like a perturbation by a negative control guide RNA. And so what I'm doing here is I'm plotting the p-values that the original authors obtained by pairing the negative control guide RNAs with the genes. And what we see is that the p-values exhibit very severe inflation. So we'd like to see these dots, so each point here corresponds to a negative control gene pair. These points should lie along this diagonal line that would illustrate correctly calibrated p-values. But what we see instead is severe p-value inflation. So basically the p-values deviate substantially from the expected uniform distribution under the null hypothesis. So why is this the case? Well, there are sort of two, I would say, core challenges in the analysis of these single-cell CRISPR screen data. So the first challenge is that technical factors such as sequencing depth and batch affect both guide RNA detection probability and gene expression measurement. The idea is, sequencing depth, for instance, varies across cells, and so cells that are sequenced more deeply would be, would have a higher measured gene expression, but we're also more likely to detect a guide RNA in cells that are sequenced more deeply. So basically, these technical factors are acting as confounders, traditional non-parametric approaches like permutation tests and chi-squared tests of independence are not robust to the existence of these confounding technical factors. So sort of standard non-parametric approaches are not going to work for these single-cell CRISPR screen data. The easiest way to correct for technical technical factors is to include them as covariates in a parametric regression model. So for example, you could include sequencing depth as a covariate in a negative binomial regression model. However, this is also problematic because the single-cell CRISPR screen data are very noisy, and it turns out that parametric models tend to not fit very well to the data. So it turns out that we're kind of in a challenging situation here. Basically, non-parametric approaches are not really going to work because of the existence of confounding technical factors, but parametric approaches also are not going to work very well because of the noisiness of the data. Parametric models don't fit to the data very well. And so we're sort of at an impasse here. And so we propose to sort of solve this problem, we propose to use a method called conditional resampling. And conditional resampling is a new statistical technique. I mean, I think the idea has been around for a while, but it was kind of formally proposed a few years ago. And basically, the idea behind conditional resampling is it enables us to adjust for these technical confounders like sequencing batch, but at the same time, it's robust to misspecification of the gene expression model. And so it's kind of solving these two core problems simultaneously. And we implement the conditional resampling approach as Scepter. So Scepter basically is a custom version of conditional resampling for single-cell CRISPR screens. I don't really have time to describe how it works, but basically It's a version of conditional resampling based on a negative binomial test statistic, and we implement several statistical accelerations to enable this to scale to like large-scale single-cell CRISPR screen data sets. So in practice, it's fast to apply to real data. Now I want to share some results on real data. So I'll focus on the Gasparini data. I don't really have time to share results on the SHE data. Well, actually, I will actually show calibration results on the SHIA data. But yeah, so basically what we're looking at here are p-values that were computed by pairing negative control guide RNAs to genes. Recall that we're under the null hypothesis here, and so we want to see the p-values sort of uniformly distributed, right? We want to see the p-values lie along the diagonal line here. That would indicate good calibration. So what we see is Scepter, which is in blue. Scepter's points lie along this diagonal line, indicating sort of correct calibration under the null hypothesis. Whereas the competitor methods, which are shown in other colors, basically demonstrate severe miscalibration. The competitor methods are a mix of some of them are like parametric methods and some of them are kind of like standard non-parametric methods. And so our hypothesis is that they're kind of not working for the reasons that I sort of explained earlier. So yeah, so we see solid calibration here, which is a nice result. The discovery set of Scepter also showed improved enrichment for biological signals of regulation. So in particular, we compared the Scepter discovery set to the discovery set reported by Gasparini et al. In their original paper. Gasparini et al. Used a kind of parametric negative binomial approach to do this differential expression testing. What we see is that the Scepter discovery set diverges substantially from the original discovery set. So in particular, among the pairs discovered by either method, among the gene enhancer links discovered by either method, Scepter and the original method only agree on about 50% of those links. So pretty substantial disagreement or divergence between the two methods. So we compared the Scepter discoveries to the original discoveries on several metrics using orthogonal functional assays. So what we see is that the Scepter gene enhancer pairs, the gene enhancer links discovered by Scepter, are physically closer to one another on the chromosome than the gene enhancer pairs discovered by the original method. Moreover, we looked at topologically associating domain data. So Scepter discoveries, Scepter gene enhancer pairs were more likely to fall within the same topologically associating domain than the original pairs. And this was despite the fact that Scepter discovered almost 100 more gene enhancer links than the original method did. And finally, we looked at ChIP-seq data. Basically, we're looking at the enrichment of enhancers discovered by either method on 8 ChIP-seq targets. And what we see is that the enhancers discovered by Scepter demonstrate greater enrichment on these 8 ChIP-seq targets associated with enhancers. And I'll just mention here that the ChIP-seq targets that we're looking at are the same ChIP-seq targets that were examined in the original analysis, in the original paper. So basically, we replicated the analysis of the original paper to compare the two methods. And what we see when we compare the discoveries unique to Scepter to those against, let me phrase that a little bit differently. Basically, to sort of investigate the differences in greater resolution, we examine the symmetric difference of the discovery sets. So what we're doing here is we're comparing the discoveries unique to Scepter against those unique to the original method. And what we see is that the differences become even more extreme. So for example, the Scepter gene enhancer pairs are less than half the distance to one another than the pairs discovered by the original method. And looking down here on the right-hand side at the ChIP-seq enrichments, we see that, for example, Scepter is much more enriched for this ChIP-seq target H3K27AC. This is probably the most important of all the ChIP-seq targets that we looked at. It's strongly associated with active enhancers. And so we see that Scepter enhancers are much more enriched for this important ChIP-seq target than enhancers discovered by the original method. And the difference is pretty substantial. And so just to wrap up and sort of think about future directions, single cell CRISPR screens, I think, will play a key role in dissecting regulatory mechanisms underlying disease associations. I just want to highlight a recent work conducted by collaborators of ours at the New York Genome Center. They developed an approach called STING-Seq, and STING-Seq combines biobank scale GWAS data, single-cell CRISPR screens, and Scepter to map GWAS hits in cis and in trans. So I don't really have time to discuss this in detail, but basically what they did is they sort of looked at this phenotype of basically of quantitative blood traits, and they identified a bunch of SNPs that are associated with this phenotype of interest. And the SNPs that they identified are all like non-coding SNPs. So these are SNPs that lie outside of genes. And then what they did was basically they ran a single cell CRISPR screen. So they targeted these non-coding SNPs, and using a single cell CRISPR screen, they were able to map these SNPs to target genes both in cis and in trans. So a very interesting study, and the analytical tool that they used to analyze the data was Scepter. And Scepter apparently was able to produce well-calibrated and sensitive results on these data. Yeah, I see sort of, interrogating the role of GWAS hits through single cell CRISPR screens as a core like application area of Scepter. And the manuscript is down here if anyone wants to check it out. But with that, I think I can wrap up. So thank you so much. Yes, the manuscript is available on bioRxiv, and the software package is available on GitHub if you want to try that out. And I think that's everything that I have. So thank you.
Thank you. I will stop my timer. You have plenty of time for questions. So we have a question from Ben. Great talk, thank you. Is it possible that sequencing depth isn't necessarily a co-founder, but could actually be affected by a CRISPR edit? For example, if the edit harms the cell or inhibits transcription somehow?
Yeah, I mean, I think that's a great point. Yes, the CRISPR edit itself might be, affecting some of these technical factors. So yeah, I guess the suggestion here is that gRNA, so I've got a DAG here that basically represents the causal relationship among the different sort of variables that we're considering. And I guess the idea here is that sort of gRNA presence might actually affect the technical factor itself. And yeah, I think that's an interesting point, and it's totally possible. I will say that these experiments are designed in such a way that sort of the cells are not overloaded with CRISPR perturbations. So to my understanding, when you sort of transduce too many CRISPR perturbations sort of to the cells in the experiment, the cells do end up dying typically. So you kind of need to be careful with the number of sort of CRISPR edits that you transduce into any given cell. So I think that the biologists have been pretty careful sort of on that issue. And so yeah, so as far as cell death caused by CRISPR edits, I'm not sure that's a substantial issue. But yeah, I think that's an interesting point, that there might be some bidirectional causality happening in this DAG. Yeah, something to consider.
Okay, we have another question. Can Scepter be used for experiments which use CRISPR-Cas12a instead of CRISPR-Cas9?
Maybe so. Yeah, I mean, so we're looking into extensions of this method to different settings. So our focus so far has been on basically CRISPR-I experiments in which enhancers or promoters are perturbed. We are currently working to extend the method to different settings, to low MOI settings in addition to high MOI settings. as well as to like different CRISPR modalities. So CRISPR-A, I agree, would be super interesting to look at. So I guess I would say there's no conceptual reason that Scepter wouldn't apply to those different kinds of experimental setups. But yeah, it's not something that we've, well, it's a work in progress, I guess, but it's not something that we've published or anything.
Okay. So there's still a couple of minutes in case anyone has another question. Otherwise, there's 1 minute in case anyone has another question. Otherwise, thank you to all the speakers of this session. And since we are at